now know what the aperiodic crystal is, upon which all life depends, and we know how it replicates. Now, in a sense, at this stage, I almost ought to apologize to you. I'm going to tell you how nucleic acids carry the heredity of life. I hope, in a sense, that most of you already know. My excuse for repeating it is that I do think it's certainly the most important discovery in biology of this century, probably the most important discovery in science in this century, and there is some excuse for repeating such discoveries, even if you already know them. The essence is shown in the next slide. I'm showing it for a molecule called RNA, not for DNA. For the purposes of this discourse this evening, the difference really doesn't matter. And that is my rather non-chemist's idea of an RNA molecule. It's a long string, and it's a string of 4 kinds of nucleotides labeled A, U, G, and C. And they have chemical formulae, and the fact that I don't remember their chemical formulae doesn't actually worry me all that much, because I was relieved to discover, reading many years ago, Jim Watson's discovery, I was going to say novel, it isn't actually a novel, his book, his account of the discovery of the structure of DNA. He admits that he and Francis didn't know the formula of adenine and cytosine and so on at the time, so I'm in good company. Anyway, these are 4 different molecules, and here are the isolated nucleotides, not all stuck together, which are supposed to be loose in solution, rather in the way that in that tube, before we put the crystal in, there were the units from which the crystal could be made. But here there are 4 different kinds of units, and the rule is a very simple one, that U pairs with A, that A pairs with U, A with U, U with A, but C pairs with G, and therefore C with G, G with C, and then all you have to do is to zip the thing together. It's a bit of a problem, actually, but that's all in principle you have to do, and then you've got not a copy of this, but a negative image of that, in the same way that a negative is the inverse of a photograph of the positive. But if we then proceed to copy this by the same mechanism, we'll get back to that. And that really is the essential process which underlies all of life. My friend Leslie Orgel, talking about this, once said that if you try to sort of trace back the history of life, back to its very origins, things sort of slough off one by one. You know, you'd use legs and arms and leaves and things like that, and then you lose cells. Ultimately, you go back, and you're left with just the one simple principle of complementary base pairing, and this is complementary base pairing. And he said you get left with complementary base pairing like the smile on the face of the Cheshire Cat. And I think that's right. It's really the root origin of the whole thing. Now, the thing I want to say about this is that one way of thinking about heredity, which wasn't obvious to us for a long time, but I think is obvious now, is to think that it's really just a matter of transmitting information. And as a result, I suppose, of the work of Wiener and Shannon at MIT during the war and others, we now have a kind of science, we have a mathematics of information, of how you measure the amount of information, how you transmit and encode information. The essence of that mathematics really rests in the notion that the amount of information in a message is simply to be measured by how many other different messages might have been transmitted and when. First sight, that sounds rather an odd idea. But you can illustrate it like this. You have your wife, your husband, your boyfriend, your girlfriend is in prison. And you receive a letter from them. And the letter simply says, I am well. How much information does that message give you? Well, it depends upon how many other messages he would have, or she would have been allowed to write. If the rules of the prison are that if you write a letter, it has to say, I am well, and that's the only letter you can send, then in fact the letter gives you what technically we would call one bit of information, and one bit only, that you're alive and not dead. If, on the other hand, it's a nice, modern, open prison in which you're allowed to do whatever you like from drugs downwards, then you could have written anything. Now the message, I am well, you could have written, I've got an attack of shingles, or, I'm going to break out tomorrow, or all sorts of things. The message, I am well, gives you a lot of information. So we measure information in terms of the number of different messages we might have received. And when it comes to the information in nucleic acid, the amount of information is simply 2 bits per nucleotide. It's simply the number of different nucleotide sequences of that length that might have existed other than the one you've actually got. Now, that's in a sense a digression, but I want to persuade you that for much of biology is today really permeated by the notion of information, and perhaps the simplest way is to remind you, or tell you, I'm not sure which it is, of some of the terms which are standardly used in molecular biology today. And I'll read them because I won't remember them otherwise. Translation, transcription, editing, code, message, reading frame, proofreading. All those they're not loose analogies. Those are all technical terms. They have quite precise meanings. I was reading a book recently, published by a friend of mine, I won't mention his name, it would be quite unfair, the other day, which contains the statement that bacteria don't have proofreading. Now, proofreading has such a precise meaning that I can assert that that statement is wrong. Bacteria do have proofreading. I say that not because I want to get at my friend, it's a very good book, but because I want to indicate that this is a word which doesn't have a loose metaphorical meaning, it has a very precise meaning. You can say wrong. And not only does it have a very precise meaning, it means exactly almost what you would mean if you talked about proofreading. It means that when a cell, this is going on inside a cell now, replicates a nucleic acid molecule, having done all this pairing up, there's an enzyme that then goes along, group of enzymes goes along, looks at all these pairings, checks them, and if one of them is wrong, it takes it out and corrects it. That's exactly what you do when you do proofreading. And there's a special machinery for proofreading. So, today, molecular biology is simply about information. Now, I want to be even more general than that and suggest to you that, in a sense, the last century, the 19th century, was a century in which science discovered how energy can be transformed from one form into another. From chemical energy into mechanical energy in a heat engine, in a steam engine, and from mechanical energy into electrical energy or vice versa, in a dynamo or an electric motor. This century, I believe, when we look back at it, will be seen as a century in which we discovered how to transform not one form of energy into another, but to translate information from one form to another. Though things never happen in neat centuries, of course, and the first machine which did this, well, I suppose strictly this is the 10th machine. Let me explain. This is Edison's phonograph. It's Edison's phonograph number 10. So I guess it's the 10th one he made. What it does is to convert the information contained in some grooves on this soft metal here into sound. It's not a terribly interesting sound, but it is a sound. Right? And, well, you can work out how it's working. Now, I guess that's not all that marketable. That's 1877. 11 years later, this was on the market. This, I'm not sure what this is actually called. Oh, it's on the thing. here we are. The storage of information has moved from the metal to a wax cylinder. There's a groove on the wax cylinder. I don't think you can see it, but that's where it is. And on the label, it says, Edison's cold molded records echo all over the world. Yeah? And it does. Can you make it make a noise, Bresson? Can you hear a voice? There. That's the voice of Bill Coates.